Good evening, everyone. It's my pleasure to welcome you all. So we have one of the very nicest topic. Drug discovery is one of the topics that we need to talk. We also have a drug discovery workshop that has been planned from March 13th to April 2nd. We'll send you the link later so you can have a look at it. It's one of the comprehensive workshop with around 50 to 55 speakers that we are planning. But before jumping into today's webinar, I would like to tell you what is FABA. So for many of the veterans, you might be knowing FABA. For the new people, so FABA is the Federation of Asian Biotechnology Association, started in 2005. And one of the flagship event was BioAsia. And the main aim of starting FABA is to bridge the gap between academia and industry. So mostly through events, we used to do it. In 2020, we started something called FABA Academy. FABA Academy is all about upskilling people, whether it could be students, industry insiders. So we upskill and then try to provide them connections into the industry where they get jobs. So this is where FABA Academy today is. The webinar is from FABA Academy. Last year in February, last year in August 2022, we also launched an event called Whale Tank, FABA Whale Tank. Whale Tank is all about bringing investors to startups. That's where we launched a new division called FABA Entrepreneurships. So now FABA, with its huge network, we have chapters in around 20 countries. We operate in FABA Academy and FABA Entrepreneurship. So that's how FABA operates. So whoever is interested in working with FABA, or if you need any trainings or anything, please reach us out. We have membership models. So it's a non-profit organization. We operate through memberships and sponsorships. Please reach us out. We'd be happy to guide you through. That's about FABA. So today, I would like to start the session by introducing Rami Reddy Mutyala. So it's a pleasure to have you, sir. So in his 25-year career, Dr. Reddy has contributed greatly to drug discovery. He's one of the world's few computational method developers and drug discovery experts. He was responsible for successfully developing a highly accurate computer-aided drug design, CADD, technology intended to speed up drug discovery process. He's currently co-founded and chairman of Rational Labs. Sir, it's a pleasure to have you. Now the stage is yours. Feel free to share the screen.
Yeah. Thank you, Dr. Jagadish, for the introduction. First, I would like to...
One point I would like to add that he is the first PhD from University of Hyderabad.
Oh, amazing, sir.
Okay.
Yeah, first, I would like to thank Professor Reddy and other organizers for the invitation. So, even though I worked on more than 20 different drug discovery projects, due to confidentiality, I cannot talk most of those projects. But today I will discuss two successful projects. One is for diabetes, another one is for AIDS, using novel computational methods in structure-based drug discovery. So this is the outline of my talk. This is the outline of my talk. First, I will give you a brief introduction to drug discovery, then type 2 diabetes, and how we are going to tackle that problem using fructose 1,6-bisphosphatase inhibitors, then use of CAD methods for discovery of potent FPPase inhibitors, then use of CAD methods for discovery of a drug for AIDS, then advantages and limitations of current CAD methods, then how we are going to eliminate some of those limitations using a novel quantum mechanics, molecular mechanics based free-energy perturbation method. Then last, I will discuss CAD successes and limitations. So this is the binding process. This is the ligand in the aqueous phase. This is the receptor in the aqueous phase. During binding process, this ligand is going to get into the cavity of the receptor by kicking the solvent out. There are two approaches for drug discovery. One is a traditional drug discovery, another one is a rational drug discovery. There are two approaches for rational drug discovery. One is a structure-based drug discovery, when you know the structure. When you don't know the structure, you can use a ligand-based drug discovery. So today I will focus mostly on use of computational methods in structure-based drug discovery. In structure-based drug discovery, target is known, and the 3D structure of the target is known. In many cases, we know the complex structure. Once you know the complex structure of that lead compound with the target, we can identify the properties of the binding site, where you have a hydrogen bond donor or acceptor groups, where you have a hydrophobic and hydrophilic areas, and volume of the active site. Once you know the properties of the binding site, we can design new ligands which are specific to given target with a high potency. So this is the time frame for drug discovery and development. Time required for discovery of a clinical candidate anywhere between 3 to 8 years. Time required for clinical studies phase 1 to phase 3, 3 to 5 years. Time required for NDA submission and FDA approval 1 to 2 years. The total time required is anywhere between 7 to 15 years, and it costs anywhere $250 billion to $1 billion. So this figure, this white bars indicates investment in failed compounds. This blue bar indicates investment in successful compounds. As you can see, a lot of money is wasted in lead optimization and phase 3. Using computational methods in structure-based drug discovery, one could design new analogs of a lead compound which are more specific to a given target with high potency, which would result in number of failed compounds in phase 3. As a result, that will reduce the investment in failed compounds of lead optimization as well as in phase 3. So this is the going from gene to all the way to drug. So of course, if you start with the gene and go all these steps, that will take more than 10 years and cost more than $1 billion. If you start with the lead and go here, it costs much less money and much less time. So our cost of the project and the time for the project will depend upon where you start the project. So this is the flow chart for CAD. So before you start using CAD, so we need to obtain necessary hardware, software, and databases. Once you have all those three in your hand, first question one should ask is protein structure known? If the answer is no, obviously we have to use QSR approaches, either 2D or 3D. Using any one of these approaches, one could suggest new analogs of a lead compound using Farm-4 model. If the answer for this question is either yes or no, still one could propose new analogs of a lead compound using hydration free energies, provided inhibitors bind to their enzyme in their hydrated form with covalent coordinate bonds. So in 1998, we developed a novel method for calculating hydration free energies very accurately. Even now, that is the same method, nothing is improved. So still that is one of the best methods. So the next is if the answer is yes. So first step is, so there are several generations of the computer model. So what I mean by generation of computer model is first step under this, develop missing force field parameters. So if you take any commercial or academic package, they do have very good force field parameters for all known small organic molecules and also many of the known drugs. When you go for novel molecules, none of these packages have good force field parameters, even though they claim they have parameters for every atom in the periodic table. So obviously, user has to develop the parameters given in this equation. So bonds, angles, torsions, non-bond, if they want to get a good agreement. So I will come back to this one later. So the next step is building missing atoms or residues or loops. Whenever you have a missing atoms or residues or loops in the protein structure, you need to build those things using homology model followed by Monte Carlo or molecular time simulations. The next one is assign histidine protein states. Since crystallographers cannot assign hydrogens for the protein amino acids, user has to look around each nitrogen of the histidine and assign the proton based on neighboring residues, such that they would pick up good optimal interactions. For example, you have this lone pair here. If you have another carbonyl oxygen here, you want to put a proton here, such that they would pick up optimal interactions. Same thing here. When you have NH here, if you have a NH2 group here from amino acid, then you don't want proton here. So you need to take out. So you basically user has to look around all the neighboring residues and decide where the proton goes, epsilon or delta or both. Then next one is orient side chain for SN and Glen. So when crystallographers solve the structure, they cannot distinguish electron density between nitrogen and oxygen. So very often they assign the nitrogen and oxygen group randomly. But user, before they do the structure for drug design, they have to look around this nitrogen and all the neighboring residues and rotate these groups such that they would pick up optimal interactions with the neighboring residues. For example, if you have NH2 here and NH or NH2 from side chain, then you don't want protons here. Then you need to orient this oxygen such that this oxygen would pick up optimal interactions with NH or NH2. So you need to take care of all those things before you start using that structure for drug discovery. Even though there are some programs which automate these things, but I would recommend even you can use those programs, but at the end, you go and check all these residues and make sure they're all oriented properly. So if you don't know the lead, you can use one of these three methods. The de novo molecular design, searching databases, and HTS. So for using these methods, you need to have the structure of the target. Even this also, you need to have structure of the target. For this one, you don't need. So today, I'm not going to go through all the details of these methods because I don't have that much time. So here I'm assuming already I have the lead so that I don't have to go and explain all those details to get the lead. The next one is type 2 diabetes, using fructose 1,6-bisphosphatase inhibitors. So based on 2020 survey, there are more than 450 million people who are suffering with diabetes, and is expected to grow to 580 by 2030. So India, 100 million. This is official number. Unofficially, it could be much more than. I was told in Hyderabad that 1 in 3 or 1 in 4 they have people are suffering with diabetes. In China, 90 million. That is official, but unofficially that could be much more than that. In the US, 5 million. That's about 10%. This is fairly accurate number. So it takes more than $100 billion to treat these patients' direct or indirect effects. So these are the oral available drugs for treating diabetic patients. So unfortunate thing is, in spite of all the oral available drugs, still there are more than 50% of the diabetic patients who are not able to keep up their hemoglobin A1C less than 7%. That means there's a lot of scope for improving the existing drugs as well as for discovery of novel drugs. So this is the target protein. This is called fructose 1,6-bisphosphatase. This is the lead inhibitor, adenosine monophosphate. Indication is for diabetes. So this is the structural information of this complex. This is a tetramer. Each monomer has 340 residues. Total 1,360 is a fairly large protein. So this structure was solved by Professor Bill Lipscomb and his group. So this red color is one of those monomers. As I said, is a tetramer. This is one of the monomers. This is the AMP is binding in that monomer. So this is the electrostatic surface. This green is a hydrophobic. This red one is hydrophilic. So as you can see, wherever you have this phosphatic group, you have good hydrogen bonds. And here on this ring, even though we have some polar groups, those polar groups don't have any hydrogen bonds. So again, the same thing in 2D. So we have this good 6 hydrogen bonds, and we have this 6 amino, 2 hydrogen bonds, and 7 hydrogen bonds, and here also some type of hydrogen bond. So when you started this project, we defined 2 goals. This is the lead compound, it's 1 micromolar IC50. So 2 goals, Subar. One is to convert the phosphate into non-phosphate. Second goal is to improve the binding affinity from 1 micromolar to sub-nanomolar. To achieve those 2 goals, we decided to use computational methods in structure-based drug discovery. Then flowchart for optimization of a lead compound. As I said earlier, once you know the lead compound and the properties of the binding site, we can propose thousands of new analogs of a lead compound. Just for the sake of argument, let us say I have the lead compound. I know the properties of the binding site. I proposed 100 analogs. Next, I will discuss how I'm going to eliminate 90% of those 100 analogs using these simple computational methods. Okay. Once we have proposed the 100 analogs, first thing we should do is we should dock all those molecules graphically, or just be looking graphically and conformational analysis. Anything which is going to bump with the protein, we are going to throw them away. Any inhibitor, any new analog which cost more to dissolve it, If that polar group doesn't pick up any hydrogen bonds, we can throw those things away. And also using some simple docking scores, we can eliminate 50% of those things just by using the simple tricks. The next one is for the remaining 50, we do minimizations both in complex as well as in solvent phase. So and calculate the interaction energies of the ligand with protein residues, and same thing in the solvent, interaction energy of the ligand with solvent molecules. Because when you take the total energies in minimization, they don't have any meaning. Because based on where you stop your minimization, you get one number. And also those numbers are very large. So getting differences between those two large numbers is not going to make any sense. That's why you need to take the intramolecular energy and interaction energies rather than taking the total energies. And based on those intra and interaction energies, and rank order them 1 to 50 and eliminate bottom 20. For the remaining 30, do structure-based 3D QSR by adding the same parameters plus hydrophobicity. Then you rank order them 1 to 30 and get it up another 20. For remaining 10, use free energy perturbation calculations and rank order them 1 to 10. Then you ask chemist to synthesize top scoring compounds and test for activity. Let us say we define in the 1 micromolar, and if results are promising, means if it is 10 to the power -7, that means we are going in the right direction. Then again, check the another checkpoint here. Our goal is to improve that binding affinity to subnanomolar. So even though we improved about 10-fold from micromolar, that is not enough. Then you repeat this one until you achieve whatever you defined in the beginning of project. Within 5 to 6 iterations, you may be able to get to whatever you defined. So then once you satisfy this one, take the compound for development studies. So use of CAD methods for discovery of potent FPPase inhibitors. So these are the 4 methods typically most of the companies and even academic institutions are using. One is docking and scoring. This is very crude estimate. So then when the doc was developed, it was meant to get rid of a lot of junk from the big databases. That was not meant for predicting binding affinities. That's why I call it very crude. Molecular mechanics calculations, quality estimates, structure-based QSAR, semi-quantitative, free energy calculations, it will give highly accurate results. Today, I will focus mostly on free energy calculations. So this is thermodynamic cycle for calculating free energies. This is the cycle 1. This is cycle 2. Cycle 1 is used for calculating relative solvation free energy between 2 inhibitors. Cycle 2 is used to calculate the relative binding affinity between 2 inhibitors. So this is to get this solvation free energy, you have to do one mutation in gas phase, going from inhibitor 1 to inhibitor 2. Same thing in aqueous phase. This is called delta G in aqueous phase, delta G in gas phase. The difference between those two things will give a relative solvation free energy between those 2 inhibitors. So in order to get the binding free energy, you need to do another calculation in the complex. The difference between the delta G complex and delta G aqueous phase will give you the relative binding affinity between those 2 inhibitors. One good thing about the free energy calculations is, so we can decompose that free energy into enthalpy and entropy. So let us say if you want to reduce the enthalpy just to make it a rigid, that will reduce automatically entropy contribution. If you want to increase the enthalpy, just put some polar groups that will increase. So that will help you to when you design the new analogs, whether you want to increase or decrease entropy or enthalpy. Again, this enthalpy can be decomposed to energy and volume term. So as I said in the beginning, our goal was to convert this phosphate into phosphonate, non-phosphate. And we proposed 2 to 5 to see whether we can get rid of this phosphate and come up with non-phosphate. And we proposed the 6 to 8 to see what is the role of this sugar. And we proposed the 9 to 17 to see what is the role of this polar groups here. Since there are no hydrogen bonds, so we thought we can improve the binding affinity by putting non-polar groups here. Then 18 to 20, what is the role of this 6 amino? And N7 is 21, 22 for this one. So the way we do is, before we make any new analogs, even though we know the lead compound, we want to understand which atoms are really critical for the binding affinity. If you believe something is really good, we should not touch those groups. Let us say this NH2 and phosphate groups. That's why for by making these mutations, that is going to help us to understand which atoms are really important, which are not important. If they're not important, then we can get rid of those things and substitute with something else. So that's why this exercise is very important before you propose new analogs. So before we use free energy for these predictions, we want to validate this method for known inhibitors. So the S1 is a ZMP, this is AMP, this is a fairly large mutation. So these are the calculated results. So this is a calculated one, this experimental, this is a pretty good agreement. So based on this, we thought, this protocol, whatever we use for calculating known compounds, we can apply the same protocol for unknowns. So these are the results. So wherever you see this negative number here, that means new analog is better than the lead. Wherever you see the no sign, that means that's called nothing but positive, right? So that's why all are worse than the lead. The first 4 compounds we proposed to convert the phosphate into non-phosphate, all are bad. Particularly the first one, just converting one oxygen to carbon, that was almost close to the 5 kcal. So we didn't even expect that one. So improvements are wherever you put the non-polar groups on the ring, they're all better than the lead. Even the 6 amino, it clearly shows the 6 amino is very important. We should not touch that one. Even the N7 is also very important. So when you propose new analogs, we should not touch that 6 amino or N7 or even that phosphate oxygens. So we need to keep those things until the end, because wherever you want to make changes is other groups like a sugar and that aromatic ring. So this is the lead compound. When we proposed to convert the oxygen into carbon, initially we thought, so we are gaining some desolvation here to get, we are kicking this oxygen out, and we are losing some hydrogen bond. But overall, we never thought that is going to be either bad or not. It may be pretty close to the lead, and that will help us to get to phosphate to non-phosphate. When I did this calculation, it came out about 5 kcal. I didn't even believe such a big change. I thought then nobody, even I didn't believe, even Computational, the other project team, they didn't believe that number, and they decided to synthesize the compound and test the activity. When they tested IC50, they got 2000-fold. When they saw that one, they didn't believe even the experimental number. Initially, they thought the chemist, they didn't give the right compound. So they asked the chemist to go and check all the QC data. They went back and checked all the QC data and came back and they said the compound is the right compound and the purity is almost like a 98 to 99%. Then they asked the biology guys to go and measure IC50s again. They measured 3 or 4 times and it came out pretty close to 2000-fold. So then we decided to put that project on hold until we understand why we lost 2000-fold. So that's where this computational chemistry or theoretical chemistry is going to play a very important role. Because experimental guys, they cannot explain why they lost to the 2000-fold. Basically, they can say, okay, but the given compound, this is IC50. Beyond that, they cannot tell anything. So then I told them, give a couple of weeks time. I will explain why we lost 2000-fold. Then I went back to the computer lab and started looking at the geometries around this carbon and this oxygen. I noticed significant change in the geometries when you convert the oxygen to carbon, which resulted repulsive interactions with this phosphate versus the neighboring residues, and is causing a major loss in the binding affinity. So that was good. I was able to explain why we lost 2000-fold. So that is not the end of that problem, right? Our goal is to continue this project with the new ideas. Then I started thinking, what is the best way to get rid of this repulsive forces? Then I decided to design from C8 position. So when I designed from C8 position and connected the phosphonate from here, I didn't see any repulsive interactions. So based on these observations, I went back to the project team meeting. I presented my observations. They liked all whatever I presented. Then they asked me, what is the optimal linker between the C8 and P group? Because that would help them to understand, to put either 2 atom or 3 atom, what is the combination of those atoms. Based on their request, so I went to the, I went and I did some calculations. So by putting 2 atoms, the distance C8 is this one, 2.5 kcal. When you put 3 atoms, this is the energy. When I put 4 atoms, this is the energy, like this. So finally, so when you have 3 carbons, minus, when you have oxygen in the middle, this one, when you have the ring, pyranal ring, that is, so based on these results, the chemist went back and synthesized some compounds. So when they synthesized this compound, they got 100 micromolar. Of course, we started with 1 micromolar, it went to 2000 micromolar. Now we are coming back. It is not better than where we started, but at least we are coming back in the right direction. So based on this information, I requested Professor Bill Lipscomb to solve the X-ray structure with this new analog. So they solved, and this is the structure. As you can see here, this phosphate has 6 good hydrogen bonds. This amino has 2 hydrogen bonds. This one has, so these hydrogen bonds there. But you don't see any hydrogen bonds around this sugar, and you don't see any hydrogen bonds around N3 and N1. When I saw this structure, I thought there's a lot of scope to improve the binding affinity by making changes on sugar ring as well as on this ring. So based on this structural information, I went and make some calculations by putting different groups on the R group under the linker. So again, here, wherever you see this minus sign, that is better than the lead. Wherever you see the positive, that is worse than the lead. So as you can see, when you put all these non-polar groups here, that is getting better and better. So based on those things, the chemists, they went back and synthesized some compounds, and with this neopentyl and 2,5 pyranal ring, they got 0.8. So now we're slightly better than where we started. We started with 1 micromolar, now we got 0.8. Okay. So then, as you can see here, there are no hydrogen bonds with the N1 and N3. So we wanted to get rid of those things. Then again, I started putting different non-polar groups on N1 and N3. And again, when I put those things, I noticed some improvement. So based on these things, they went back and synthesized some compounds. And from 800 nanomolar, this became 15 nanomolar when they put the non-polar groups on this ring. And with some further optimization, so they got this compound. So this is where we started. Then we went to this 2000 micromolar. Then we got this 60 nanomolar. So when you overlay this AMP versus the new compound, as you can see, this phosphate and phosphonate both are going in the same binding packet, and this non-polar interactions are picking up. That's why we improved this one. So again, in the crystal structure, you can see good hydrogen bonds, and this is all hydrophobic area. So this is the new compound. So this is where we started 1 micromolar. This is the final compound with the 16 nanomolar. Using X-ray crystallography, CAD, chemistry, and biochemistry, we're able to go from here to there by synthesizing about 800 molecules. The second project is AIDS. So this is the target protein. This is the lead compound. Indication is for AIDS. So here also we wanted to validate that method before we use for unknowns. Removing valine is a very large change. Particularly, it is not at the end of the inhibitor, somewhere in the middle. It's not middle, at least. So whenever, whatever the changes you are making, if they are at the end of the inhibitor, it is easy to make. If they are not at the end of the inhibitor, it is very complicated. So it was a very large mutation in those days. Even now, it is very large mutation. So, but we were able to calculate relative binding free energies between inhibitor 1 and inhibitor 2 very successfully using free energy perturbation methods. Then we published that work in the PNAS because that was the first calculation on HOA protease and also very large mutation. Nobody was ever tried to calculate that type of thing in those days. Even now, I don't think anybody can do that type of things because we have to develop a novel method for calculating those things. That is called a dual topology method. So we have to define topology for this one and the remaining part one. We go to the one topology to another topology. It was very complicated. We had to spend a lot of time to develop that method. So again, here, these are the calculated results, these experimental results. We got pretty good agreement. So Since I don't have much time, I'm going to skip these things and show you the final drug. So again, going from this starting compound to the final drug. So in those days, using all this technology of X-ray crystallography, CAD, chemistry, and biochemistry, so we synthesized about 1,000 compounds. So in all cases where experimental data is available, predictions based on FEP calculations are correct. FEP method combined with X-ray crystallography, medicinal chemistry, and biology contributed in the discovery of clinical compounds, phase 2 and phase 3, and preclinical compounds for type 2 diabetes, and one marketed drug for AIDS. The calculated results are good examples of successful application of CAD paradigm in drug discovery. Next one is advantages and limitations of current CAD methods. So as I said, docking gives only crude estimates. Molecular mechanics gives qualitative estimates. QSR methods, semi-quantitative, free energy calculations will give quantitative estimates. So limitation is it doesn't matter which method you use. All methods require force field parameters for both inhibitor and protein as well as for solvent. And your results will depend upon how accurately you develop those force field parameters. So next I will discuss how we are going to get those parameters. So then novel quantum mechanics, molecular mechanics based FEP method for drug discovery. So here again, I'm going back to the first slide. So inhibitor in the aqueous phase, receptor in the aqueous phase. During binding process, that will get into the cavity of the receptor by kicking the solvent out. And also we can measure the binding constant for this inhibitor. Same thing we can measure for inhibitor 2. If you take the ratios or logarithmic ratios of these two, they are directly related to the Gibbs free energy. If we can estimate this, if you calculate the Gibbs free energy without making any approximations, that would become a gold standard. Even experimental has to calibrate the experiment using this method. Unfortunately, we cannot solve the Schrodinger equation for more than 1 electron. Obviously, we have to make approximations. Approximations in methods, approximations in force field parameters. Next, I will discuss how the results will depend upon the method, assuming we have excellent force field parameters. So as I said earlier, these are the 4 different methods. Here I'm assuming I have excellent force field parameters for all these methods. How results will depend upon each method. So why I'm calling this is crude is, so this is the Gibbs free energy, enthalpy and entropy. Again, enthalpy can be decomposed into energy and volume term. And again, this energy is energy in the aqueous phase, energy in the complex phase, energy in the aqueous phase. So in docking and scoring, they ignore this entropy and they ignore this volume term and they ignore this aqueous phase. When you ignore all those things, and also even for getting these energies, they get based on very crude parameters. So you can imagine what type of results you get when you ignore all the contributions. So same thing with the molecular mechanics. They include this aqueous phase contribution. So that's why they get only qualitative estimate. Whereas in QSR methods, implicitly they include this entropy, implicitly all those things. That's why they get semi-quantitative. In free energy, everything is included. So only issue here is how long you do the free energy calculation to get convergent results. That all depends upon the computer time. Okay. So this is the accuracy versus CPU time. Docking is very fast. Accuracy is very low. Molecular mechanics is slower than docking, but the accuracy is better. And also accuracy is better than docking. QSAR slower than docking, accuracy is better than molecular mechanics. Using conventional free energy methods, so getting the parameters for each inhibitor, so this is much better than these programs. And when you use the quantum mechanics, molecular mechanics based, that is even better. Then in the future will tell who is going to implement everything quantum mechanically. Right now, we are treating inhibitors quantum mechanically and solvent and protein molecular mechanics. So the next one is how we are going to get those parameters. Earlier slide, we assumed we have excellent parameters. How we are going to get those excellent parameters? Because all these programs require parameters. So for solvent, we use molecular mechanics. They are well established. For protein, all 20 amino acids and 4 DNA bases, they have very good parameters. We don't have to worry. For inhibitors, we need to develop parameters for each one. So based on which is given in this equation. In order to develop the parameters, one has to have knowledge of quantum mechanics as well as experience in developing force-field parameters. Even having knowledge of quantum mechanics and experience in developing parameters, when you plug in all those numbers, if you make even one sign wrong, you can get wrong results. So this is complicated, but you got to develop these things manually for each and every inhibitor you propose. But that takes a long time. And how we are going to solve the problem, I'll discuss next. In those days, we developed all these parameters manually, and we got very good agreement. And we got a couple of JAX papers in those days, just developing those parameters manually. But our goal is, because to develop these parameters manually, based on the complexity of the molecule, it will take anywhere 3 to 6 months. In those days, nowadays it is not going to take that long. Even now, it is going to take quite a bit of time, but not 3 to 6 months. But how we are going to solve the problem, I will discuss in the next slide. So our goal here is use the solvent, molecular mechanics force-field parameters, which are well established. For protein, molecular mechanics well established. For inhibitors, we want to treat inhibitors quantum mechanically. So when you treat inhibitors quantum mechanically, we don't have to worry about parameters. Automatically, quantum mechanics will take care. So we implemented 3 different methods in this package. One is AM1 for partial atomic charges, AM1 for getting gradients, molecular mechanics for solvent and protein. Second approach is ab initio for getting partial atomic charges, and AM1 for getting gradients, and molecular mechanics for solvent and protein. So again, ab initio quantum mechanics for getting partial atomic charges as well as gradients. It takes longer computer time. Molecular mechanics for both solvent and protein. So based on your computer resources, you can decide whether you use this method first or second or third. If you have more computer time, you can use third one, you get better results. So we validated these methods for solvation free energies, and also we validated this one for calculating relative binding free energies, and also validated for understanding anomalous hydration free energies of amino amide. We got very successful results. So the for small molecules, so in olden days when they developed the free energy methods in the mid 80s, they spent at least a few months to get the parameters. This is the results they got. Here, I was able to set up this calculation by spending 10 to 15 minutes of my time. So in every single case, I got a very good agreement using new method rather than older conventional methods where they spend 2 to 3 months to get the parameters for each molecule. Again, same thing for relative binding free energies, we validated, we got very good results. So here is a large mutations for every single case. You can see here, conventional method, this new method, and all, we got excellent results. So this is the historical perspective of the computational chemistry. So accuracy, method, time period, and users. Why I said 50 to 55% is, if anybody comes to me and ask me which one is better. If somebody comes to me with 2 molecules and if you ask him to predict which one is better, just tossing a coin, right? Already 50% answer is in your hand. You don't have to do anything. So in olden days, the professors, they used to explain to students using ball and stick models. That was 1960 to 1980, mostly academic users. Later on, industries and even academic institutions, they started using semi-empirical methods. That accuracy went up to 55 to 65. So even now, some companies and academic institutes are using these methods. 65 to 75, docking, molecular mechanics, and QSAR. Most drug companies are using this one, and accuracy. So when you use the molecular mechanics special free energy methods, so the accuracy went up from 75 to 90. There are only few industries are using, mostly this is academic exercise. This accuracy went to 90 to 95% when we implemented quantum mechanics for all the inhibitors and molecular mechanics for solvent and protein. So right now we are the only one who has this one. And even though we developed this method in 2004, still nobody is able to catch up this one. It is not that easy for them to implement, because Dr. Chandra Singh, he spent 3 years of his time to implement them. Actually, one of the world experts in this technology. So only time will tell who is going to implement everything quantum mechanically. So these are the publications related to development of this technology. The next one is flowchart for clinical development. Once you identify a clinical compound using those methods, so you can do preclinical studies. If results are promising, go for phase 1, which is mostly safety and safety dose. Then if results are promising, go to phase 2, looking both safety and efficacy to 300 to 500 patients. The results are promising, go to phase 3, looking both safety and efficacy, as well as comparing with existing drugs, doing 1,000 to 2,000 patients. If results are promising, you file for NDA, and they will decide whether it can be approved or not. Even after FDA approval, still you have to do phase 4 studies for long-term effects. So these are the drugs which were discovered with the help of the CAD. It doesn't mean they pressed the button and got the drug, but the computational chemistry and structural X-ray crystallography played a very important role. So this is the structure, and this is the disease, protein target, and company. So these are all aids. This is the virus-receptor, the one who discovered at Agaran. Now this is marketed by Pfizer. So these are the different indications, different drugs. So recent survey indicated that there are more than 420 drug candidates discovered with the help of CAD methods, which are undergoing at various stages of clinical development studies for indications such as cancer, diabetes, and viral diseases. And only time will tell how many of them are going to get into market. So these are the people who helped me to get to develop this technology and also for doing these calculations. So Professor Appara is here, Professor Reddy is here. So with this, I can stop here, and we can move on to other talks, and if you have questions, we can take either now or later. Thank you. Hello.
Great, sir. Great, sir. Thank you so much. I think you have condensed the years of wisdom into 30 minutes. I can imagine.
It was a two-hour talk. I skipped a lot of things.
No problem, sir. But I am sure there will be lots of questions for the audience. But we'll keep it for later.
Yeah,
so
sure.
We'll have a panel discussion now. Yeah, okay, great. Yeah, great. So now let's move on to the panel discussion. So I would like to introduce our moderator first, Dr. Aparoy. So Dr. Aparoy Polomaraseti works as a faculty of biology at the Indian Institute of Petroleum and Energy, Visakhapatnam. He received PhD degree from the University of Hyderabad in 2010. He later served as a postdoctoral fellow at the University of Maryland, Baltimore, and assistant professor in Central University of Himachal Pradesh, Dharmashala. His research interests are computer-aided drug design, molecular modeling, and simulations. Currently, the major focus of his group is on arachidonic acid metabolizing enzymes and cellulases. Aparay sir, welcome.
Yeah.
Great. So I'll also introduce to the next panelists, and then we'll have Aparay sir, you take care. Okay. So now I will introduce Professor Prasad V. Bharatam. So Professor Prasad V. Bharatam is Professor of Medicinal Chemistry at NIPER, National Institute of Pharmaceutical Education and Research. His area of research include quantum medicinal chemistry, pharmacoinformatics, synthesis of computational design molecules, especially anti-diabetic belonging to class PPAR gamma agonist and bigonides, drug delivery using dendrimers. He received the IBM Faculty Award, Fellowship of Royal Society of Chemistry, London, and Chemical Research Society of India, a medal, and among others. Welcome, sir, Professor Prasad V. Bharatam. Yeah, great. And next, Dr. Ajit Roy received the PhD degree in chemistry from the IIT Kharagpur. He worked as a research associate at Stony Brook University, New York, USA, and then joined TCS Research, where he's currently leading the AI-based drug design initiative. Ajit has research interest in diverse topics of computational chemistry and biology. They include development of computer simulation methods to understand rare conformational change of biomolecules, development of network-based approaches to understand disease mechanism. Currently, the major focus of his group is to develop machine learning methods for discovery and development of novel drug molecules. Welcome, sir.
Thank you.
Sure. Aparay sir, please take over now. Yeah. The show is yours.
Good evening, everyone. To start with, we'll briefly discuss in 5 minutes. Each of the panel expert will just take 5 minutes to introduce our work to you. Then we'll open for questionnaire. To start with, I think Ram Reddy sir has clearly explained how advanced molecular dynamic simulation like free energy perturbation method are accurate in estimating protein ligand binding affinity. And sir has clearly mentioned that the fast virtual screening process, they suffer from inaccuracy. And one of the main reason for the limitation of these methods is because they can't answer the protein flexibility. Usually, in this fast process, you don't consider flexibility. And also there's a limitation, they don't consider the structural waters. And when you talk about protein ligand interactions, like it was proposed by Koshland, right? Induced fit metal, when the inhibitor comes close to the protein, then it causes a dynamic change in the structure of the protein that it can accommodate the ligand. And what happens is in most of the proteins, there are cryptic sites. So there are some small pockets close to the active site, which open when the inhibitor comes closer to it. And so one of the limitation of these fast methods is we can't explore these cryptic sites or small pockets which are closer to the active site. And like sir was mentioning, in the main advantage of free energy perturbation method is there's an entropy component, entropy enthalpy shift can be taken into account. And when you talk about these fast screening methods like docking, docking is widely used throughout, because that's the only way where you can cut, you can shortlist small compounds, the potential compounds from large chemical databases at faster pace. So the entropy is one of the main challenge in the fast method. So in my postdoc with Professor Alexander McCarroll, I worked on a co-solvent simulation method called as SILKS. It was developed by his lab. It is site identification by ligand competitive saturation. So I'm just briefly, I'll summarize this method in 2 minutes, then we'll go to the other experts. So when you talk about co-solvent simulation, like in normal molecular dynamic simulation, you place the protein in a solvent and you simulate it. You try to see the changes in the protein by means of Newton's law equations of motion and by using a force field. In co-solvent simulation, what we do is we typically place the protein in a solution which is mixed with other co-solvents. So when you talk about other co-solvents, these co-solvents are solutes which are small, like small organic molecule, which represent different functional groups. We know that in a protein, there are different functional groups, and even the ligand will have different functional groups. So we use different small molecules. Like if you use propane and benzene, you are considering apolar. So you use different functional groups to address hydrogen bond donor, hydrogen bond acceptor, charged, positively charged, negatively charged, and apolar groups. So you place these small molecules along with water, and when you do molecular dynamic simulations, these small molecules will act as probes, and they'll go and settle, and they'll go and distribute in the place where there is affinity. So in that way, we map the whole protein structure. We can identify what are the hotspots for different functional groups. And in the method developed by Alexander McCarroll, so not only by identification of maps, they also gave it a quantitative form, wherein the frequency distribution of each interacting group is calculated, and also the energy that is associated with that particular functional group is obtained by Boltzmann transformation. So I have used it on, I work on arachidonic acid metabolizing enzymes, and we have used on 5-lipoxygenase, and we have some positive results. There is a recent report that there is allosteric binding site of 5-lipoxygenase. So when we got these hotspots, when we used these probes, we identified that there is another binding site. So it was we could explain the experimental results clearly, and also we could also predict the activity quite accurately. Like it was much better than the fast methods. So this is one method which we could use for virtual screening and address the limitation of some of the fast screening methods. So that's what I wanted to summarize. So along with this in our lab, we also right now we are working on rescoring and developing specific scoring functions. We know that most of the scoring functions in these fast screening methods, they are developed by using some particular kind of drug target or types of drug target. But when we use a particular drug target where there is a lot of experimental information that is available, then I got an idea that why don't we develop a specific scoring function? And so right now we are doing it for lipoxygenases. We have funding from ICMR for that. And we are carrying out that method with the idea to identify new molecules. So With this, I'll pass it on to the one of the other expert here, Professor Bhartam.
Very nicely summarized. Can you hear me?
Yes, sir, you're clear, sir.
So it's nice presentation and nice summarization, and we are at crossroads with reference to the structure-based or ligand-based design. Then the
Sir-
experimental people.
Sir, the voice is not so clear, sir. Maybe microphone, if you put it near to your mouth, yeah. Okay.
I have this.
Okay.
I hope it is.
Yeah, it's better now, sir. It's better now. Yes.
Okay. Now the point I am trying to tell is about the bridge between the computer approaches and the organic chemistry laboratory work. So it is time to that organic chemistry people should trust the methodologies and start adopting the approaches to synthesize the computationally designed compounds. Most of the organic chemists want to synthesize natural products which are extracted. Okay, nature has synthesized, we want to find a method for that. Yes, whereas in the case of the CADD, CADD has gone through lots of rational approaches. So it has matured into a system that predictive my final compounds are indeed worth taking it to the laboratory, not just leave it thinking that, okay, this is all theoretical work, this is all just some kind of mathematics behind it. But there is a lots of chemistry here, and that's what Dr. Rami Reddy's lecture has proven, that there are so many fundamentals and there is so much application, and the art of the application. The art of computer design he established, and he showed it in front of us today, and I'm sure similar value system will be adopted by many CADD people. Yes, there is a catch. A set of software vendors, they always want to say that my software will give you the drug in 2 days by just clicking a couple of buttons. That is not true. Professor Reddy just now established that there is a lot of homework, and then that homework does indeed provide results. And that's what we are experiencing in our lab also. We have been able to design several anti-leishmanial, anti-malarial, anti-TB, as well as antibiotic drug discovery. And we have taken them to organic lab, and we are able to prove them in vitro in vivo studies. Yes, certainly there, that is where industry participation is required. We are open for that. So there is success story. There are several success stories. There are still some skepticism, and it is time that the collaborators in the drug discovery should remove their skepticism, and they should start trusting the computer drug design. Pass on to next panelist.
Dr. Arijit, can you please?
Okay.
Yes.
Yeah, so thank you first, all the organizer, for inviting me. So my current research focus is slightly different. So I focus on the de novo drug design, and I use one of the technique which popularly people call as AI-based drug design. Whether my voice is clear?
Yeah.
Okay, great. So the first question one can ask me that why AI, and what is the advantage of AI? So for that purpose, I mean, I can answer it in this way, that there is a lot of data that chemistry and the biology has generated during so many years. Now, AI methods or ML methods are very good in understanding the hidden pattern from the data. And it can actually answer many of the question in a different way, and some of them can be advantageous. For example, Dr. Reddy showed in his presentation the flow chart of the drug discovery. So once the target is identified, once there is a well-defined hypothesis, we know the target. And then one of the popular approach that people use is a high throughput screening. And for that purpose, they need a library of molecules. Now, the number of molecules in this library of molecule is very limited, maybe million, maybe billion or so. On the other hand, that the number of chemical that is synthesizable, that is possible, that number is huge, around 10 to the power 60. And no one has ever explored that chemical space. Now AI can actually explore this huge chemical space, but how it can do that? So what we do, we represent the small molecule. The small molecule can be represented in a various way, maybe 3D, 2D, or even in a 1D. The 1D representation that we use here is known as smiles. Now this smile has a very well-defined grammar. So when there is a well-defined grammar, you can actually use the existing natural language processing algorithm. So you can take all the molecules, let's say from zinc or CHEML, which has a billion of molecules, 1.6 billion in CHEML, and take those molecules and learn the grammar of the small molecules so that you can actually explore this huge chemical space which no one has answered. The other thing is that once you identify the lead compound, the problem is that many of the molecules are lost because they not satisfy some of the toxicity and various physicochemical properties or many other properties. So we gradually start losing the molecules. But what AI can do, so one part is the generative AI that I just explained now. The other part is that we also predict various properties. It can be log P, the molecular weight is very simply, the user can specify what is the molecular weight within the range that they need the drug, various toxicity, as well as the things like blood-brain permeability, which is important for central nervous system. So what AI method can do, it generate the molecule, at the same time it predict the property. And what it do beautifully, I mean, compared to the traditional method, any other traditional method, is that it can do on the fly property optimization. And the other part that we do is that we are focusing more on the explainability. So for example, there is a molecule. Now, which fragment of the molecule is important for certain property? Let's say blood-brain permeability. And if we have those knowledge, maybe we can change some of those properties. And the other thing that we also take care, because many of these molecules are new, so we also take care about the synthesizability. We are coming up with a method which can actually say the forward synthesis pathway of some of these molecules during the structure-based drug design. So that is how I can explain my current focus. The other part we are discussing mostly is on the structure-based and the molecular dynamics and the free energy perturbation type of method. So I would like to say, and also Aparay mentioned that those static residues at the active site, and that is the reason we need to understand the conformational dynamics in a larger way using things like MD simulations or MC simulations. And now with the advantage of hardware, we can do things what D. Shaw did 7 or 8 years ago. So now everyone has access to GPU machines. So we can run longer simulations. Maybe 100 nanosecond is nowadays very common. But at the same time, we should remember we can still not see the actual event that should happen, even using MD simulation. And that is the reason there is a need for alternative rare conformational sampling methods like transition path sampling, more advanced method compared to the umbrella sampling, and better free energy method. So that's all. Over to you.
Yeah, thank you, sir. Artificial intelligence is playing a very prominent role in drug discovery, and it has become an integrated part of drug discovery process. And so, before opening the platform for the participants, I would like to ask maybe 2 questions for the panel experts before we pass it on to the participants. The first one is like, Sir Arjit, sir, like one question for you: whether this AI-based drug design, it's one of the most important topic of discussion in drug discovery process these days. So, is it a hype or is it a hope?
Yeah.
And how it can impact the routine drug discovery process?
Right, very important question. I mean, I would like to say that one should not think that AI or ML can solve all the problem. It cannot do that. So I already explained to you that what are the advantages over the traditional method. And the data can always be a challenge, although it's a data-based method. So when someone is building a model, they should remember using this data, what answer we can give. Unless we are sure about that answer, I think it's very difficult, and that's the pain I have. I have that pain because so often I see some academic paper where people are looking at the accuracy of the method. 95%, 96% accurate, the model are 95% or more than that accurate. But often they cannot explain the right thing. So one should be very sure what is the question that can be answered with that data. But there is a lot of advantage over the traditional approach. So we should think it as another approach which can solve many of the problems. And whatever the nature of the problem, one can use different method. In the early days, people used molecular dynamic simulation. We are still using it. AI has came, and maybe few years after, quantum technology will come. I mean, it's still in the very nascent stage. I'm not talking about the KMM thing that Dr. Reddy is talking. I'm more talking about the quantum technology. But one should identify the domain people, that the domain knowledge should be there. So we should keep that. And if we keep that, if we can understand the question, whatever technology will come, we can adopt that. And based on the problem statement, we can consider any of these techniques, because each of them has advantages and disadvantages.
Yeah, thank you, sir. And I have one question for Ram Reddy, sir.
Okay.
Sir,
Go ahead.
Sir, like how important is visual inspection? Because most of the people who work on this, they usually they get the structure, then immediately they load it on some program, or they'll continuously keep doing it. But how important is to look for the structure, the visualization of the structure, and what information is very critical, so that there are many students here, they'll get very important information how to do things.
Yeah, that's a very good question. Particularly when you use the docking, and people they claim, okay, I predicted this one, this is my score. So before they present those results, whatever the top scored molecules, whatever they got, they should go and look on the graphic screen.
Yeah.
So whatever you are seeing on the graphic screen should make some sense and correlate with your numbers.
Yeah.
So if you are seeing some repulsion interactions on the graphic screen, and if you see some good results in the scoring, that means something is wrong. Okay. So whenever your visual inspection correlates with the numbers, then only you should go and present those results. Otherwise, you should go back and check those things. Because most of the time now, when you use the docking program or minimization, those things, they mislead you. Even you get right number, you get the right number due to wrong reasons.
Yeah.
So even when I was working with other companies, so I used to have a big group. So my computational chemists, they come back to me and say, okay, these are the results I got. Then I won't go and check everything, whatever they did. So whatever the top scored molecules, 10 or 15, I go to the computer graphics and look them one by one. If my intuition correlates with the numbers, then I say that we go and present. If it doesn't, then I will ask him to go and repeat that experiment or that calculation. Okay. Very often, this approximate methods, you get wrong numbers. Visualization of the final results is final success is very important if you want to propose correct molecules.
Yeah. And like continuing the question, like sir, when we look to the ligand structure in the protein active site, like how we always try to insert a new interacting group so that the free energy will come down. That's the whole idea. But how important is to look for whether there is any non-interacting group in the ligand?
Yeah, that's why so when I presented my results on FPPS, so first we went back, look at the graphics. When you look at the graphics, that's where I got the ideas, not based on my calculations.
Yeah, okay.
See, when I look at it graphically, I notice the structural change when you go from oxygen to carbon.
Yeah.
In the calculations, we didn't even expect that one.
Okay. Yeah.
And also, when you look graphically, where you have hydrogen bond donor or acceptor groups or hydrophilic areas, and if you see some area which is not picking up any hydrogen bonds or any hydrophobic interactions, you can get rid of those groups and replace with something else which can pick up a better interactions.
Okay. Yeah.
So I think you were talking about the lead optimization stage, but if someone is screening a large library, is there a way they can also take care apart from the screening score?
Okay.
Because visualization is not possible over there, right?
Yeah.
No, only the top molecules, not everything.
Only the top molecules I'm talking about, like.
Because the DAC originally, when they developed in the early 80s, that was meant to get rid of a lot of junk from the large databases. So the TAC counts, whoever developed that first TAC program, so he used to give that program for all academic and even industries in those days free of cost. In 1988 or late 80s, he was searching some databases. He found one hit for HIV. In those days, that was a big story. He was everywhere on every national channel because they thought he discovered a drug for AIDS just for getting a hit. After a few years, he didn't get anything out of that one.
Yeah.
Then in the beginning, he used to give that DAC program free of cost. After he got that hit, he started charging for every institute.
Yeah.
He became very popular because of the DAC program.
Yes.
Originally, it was developed, just like a shape-based docking. Nothing, you didn't even implement some energies there. Based on shape. If it doesn't fit in the binding set, just get rid of those things. Later on, you started incorporating energies and other things, some flexibility and all those things,
Yeah, they just looked at it as a molecular sphere,
whether it fits in the binding set or not. Just to get rid of junk. That's it. Then later on, they added the flexibility and some energies and other parameters. And also other commercial package guys, they took that original code from him and they implemented other things.
Yeah.
Original DAC was developed by TACons.
Yes, sir. Thank you, sir. Bhartam, sir, I have one question for you. Maybe I want your explanation on this. Like usually when we talk about, usually talk about designing drugs for known drug targets. My question is, can we do the other way? Can we identify a drug target for a drug using these computational approaches? Sir, can you unmute, please?
I'll address that problem if you permit me, Dr. Purai.
Sir, after Bhartam, sir, just you can.
Yeah.
Okay, that is a very interesting question. It is possible in principle, but in practice, it is it will take so much more time to do. So not it is being practiced by everybody. Few people are doing it across the world. But before I even attempt the answer to your question, I want to add more to what Rami Reddy mentioned.
Yeah, please, sir.
It is 3D. 3D, when we visualize what we are visualizing, it is a 3D.
Yeah.
We should actually go out of our 2D domain. 2D domain was convenient for us when we write on paper and with pen, or we draw on the screen or anywhere. But now we are thinking in 3D. We should have that ability to think in 3D. Then we have to cultivate that habit. If we successfully cultivate that 3D thinking, it is possible to achieve high success. This is what has been practiced by many people. Those all the successful people, in my opinion, and in my observation, all the successful people developed this habit of thinking in 3D. It's not just leaving it to 1D or 2D. So this,
Yeah.
sometimes I feel pain when I see any pharmacology or any biology related paper. They don't even show the 2D structure of the compound. They only simply write the name and simply go ahead and complete the entire paper. So much has been discussed. Drug discovery is discussed, but did not even show the 2D structure. So
Yeah.
it is important for everybody to think in 3D. Like I see many of our institute students, for example, a student spent 5 years with one drug molecule, that is tamoxifen. His idea was to develop a better delivery system.
Yeah.
Okay, 5 years he spent, but he never saw the 3D structure of the compound. It is a pity. So, so to answer your question, that is visualization, that visualizers are, all the software visualizers are readily available today to help us in thinking in 3D. Just visualizing is not sufficient. Grasp it into our mind and then utilize it it is required for the artificial intelligence also.
Yeah.
So, will get the appropriate parameters.
Yes.
Okay, now coming to this question, can we not design the targets? Can we not identify new targets? Yes, why not? There are many methods where we need to integrate bioinformatics approaches, that is sequence similarity, sequence comparison, the scores, PMF score, and other things.
Yeah.
And then we need to integrate the computational methods and then see a combined integrative approach, that is with reference to the pharmacoinformatics. That's what we are promoting at
Yeah.
Mahali.
Yeah.
Pharmacoinformatics, that is not even sufficient. We need to correlate all the time. Whatever result we get, we need to correlate with the reported biology. We should not. We should trust our chemistry. We should trust our biology. We should integrate all those results. We are far from the workbench. A person who is working on a workbench, he may not have a greater vision. He will see a particular test tube is doing, whether his compound is getting eluted or not. He may not have time to think in a big way. Computational chemists have got the big opportunity to think about the biology as well as chemistry and then about the 3D. So I'm sure it will all give the results. Your question can be positively answered.
Yeah. Thanks a lot for that.
I just want to add a couple of more things. He has given all the answers. I just want to add that right now there is a lot of work towards creating the knowledge graph using all the multi-omics. So I think that is what he has mentioned as the integration of the data, and from that find the relationship between the disease protein, disease drug, or the drug protein. And not usually people, once they integrate it using maybe from text, from all the PubMed data, some relationship ontologies, and then try to integrate all those data into thing. I mean, the data can be unstructured, the data can be structured.
Okay.
And I think then people may sometimes often use methods like graph-based approach to find the link predictions, and these algorithms are quite popular in understanding the relationship between and finding a new target. And there are a lot of work has been done. For example, one of the company known as the Benevolent AI, I mean, they has a huge knowledge graph, and they have done considerable work on finding the target. And I think now you can hear a lot of things about the ChatGPT and the large language model. So I think that will play a disruption in this particular field to finding the relationship and finding new target.
Okay. Okay. Yeah.
Can I?
Yes, sir.
Can I come down? Okay. Yeah, your last question is a very important question. If Professor Rami Reddy remembers, when he was introducing me to C.R. Rao Institute, I asked him this question: how do you choose the target? choosing the biological target is important. And when we are asking is to choosing the target, whether actually the present models of metabolism on which actually we are designing drugs, are they right? This is one question which I have been examining for nearly decades. The docking experiments or all other experiments are okay. I am not competent to talk about the chemistry of those things, although I understand the limitations of that particular thing, which were highlighted by Professor Rami Reddy very perfectly. If you permit me, I'll give you three slides. I highlight actually where it has gone wrong. That will be actually the focus, and it will be a turning point in the drug design systems. Otherwise, it will be like playing a carom board as to who is the expert who is playing the striker, and then what pocket he is choosing and what coin he is choosing, whether it is actually entering into that particular target or not. Okay. If you permit me that, I'll just the first thing that is coming into the picture is top-down models and then bottom-up models. These two models have been built up based on actually in vitro analysis of these things. And these are the two models which have destroyed the entire process of glycolysis and Krebs cycle. It distorted actually this thing. In fact, 1922, 1923 Nobel Prizes, which are awarded simultaneously in October and December 1923, are the starting points of actually this thing. The hype was the application of biophysical and biochemical tools in analyzing the biological methods. That was attractive system in this thing, but in the process, they have actually boiled onto this thing. And I think at the end of 1960s, Britain Johns introduced actually computational techniques into chemical equilibria, and those particular things were taken up later, and then they went into this thing. This particular top-down model was given by this thing. Before that, I'll just go to the first decade of 20th century research, which was overlooked. And one of the two important developments that took place in Hyderabad and Calcutta in India itself during that period were ignored along with other. Now, after the Pasteur's experiments on this thing, and then Buchner's discovery of enzymes, we had actually the Harden and Young methods in which actually in vitro alcohol production by cell-free systems of yeast. They introduced two equations which are called Harden and Young equations. The equations themselves have been disputed by that time by another Nobel laureate who shared his Nobel Prize in 1929 along with Harden. And then there is a difference between this thing. Parallelly, between 1907, parallelly in Hyderabad, actually, there are two issues in this things. 1922 Nobel Prize on muscle metabolism, exercise metabolism centers around two things. Number one is thermogenesis. Number two is lactic acid production. Glycogen lactic acid theory, glycolytic lactic acid theory, which remains as a dogma even to this day, is because of that particular Nobel Prize. Now, they ignore fundamental things that, the fructose is a poisonous sweet. This was discovered in 1907 by Richard Havelock Charles within Calcutta. Cane sugar is not native to India. We got it from Polynesian islands, and fine sugar preparations were there only around Calcutta during that period. Even in Punjab, actually, fine sugar preparations were not there. Havelock Charles observed one simple observation was that, those kids, rich kids who had access because of the price variation for this particular fructose-rich cane sugar were developing obesity. This is the first indication that, fructose contributes to the obesity is one of the major observation which was overlooked by these things. Parallelly, there was also other in Sweden. So I won't go into that thing, but fructose is one of the important things. It is only after nearly a century we from 1930s, from the middle of this 20th century, people started examining that, fructose could be this thing because ice creams and other things, they were causing actually globally obesity, and they were corn sugar or any sugar which is rich in fructose is a causative factor for these things. And in this particular thing, we have century of reports also that, fructose metabolites inhibit actually the hexokinases and inhibit actually glucose uptake into the cells. It is only in the cancer cells when actually fructose metabolism is downregulated, you have actually the carcinogens. Hepatic cancers are activated only when ketohexokinases are inhibited. Otherwise, only hepatic steatosis or non-alcoholic fatty liver disease is one of the major factors in this thing that leads to the fibrosis, and then hepatic damage is a secondary issue. So fructose ignorance on actually this particular system, ignoring this particular factor led to this thing. Then comes into the picture the Warburg is the hype. Warburg effect is the hype. You take any paper on cancer, the first thing it starts while I am talking, there might be 100 papers written on cancer. There will be Warburg effect is the primary system. What does Warburg effect say? ATP is the sole molecule which modulates the energy. And how does actually Warburg effect come into the picture? He discovered NAD, NADP, and thereby he attributed that, only oxidation reduction reactions and glucose catabolism leading to the ATP production is one of the major sources of this. In the previous paper, I have forgot to tell you that, Euler clearly stated that, and even Harden has clearly stated that carboxylation reactions will suppress the fermentation process, activate regenerative process. Euler clearly stated that atmospheric oxygen uptake activates actually carboxylation reactions. It activates actually the biogenic programs, not actually the catabolic programs. Contrary to that, we have aberrations in terms of actually that insulin promotes actually the catabolism of the carbohydrate, and we have a respiratory quotient is another aberration that has come into the picture. If you look carefully into these things, you will also see that, whether it is the glycogen catabolism or actually the shift of these things, you have actually the vitamin B systems which are actually important in this particular step.
Yeah, sir. Yeah, thanks for your insights, sir. I think these are very important insights, and I think we can directly get in touch with some of the experts here. We have few questions from the participants. If you can allow, I can just take those questions.
Yeah, this is the last one I'll be giving. This thing is, unless we remodel the entire metabolism, and recent two developments, I put many of these things in LinkedIn also. Some people interested can look into that particular thing, that, the structure and function of mitochondria. VDAC has a different role from cardiolipin. There are two important papers that have come up in the recent past, one from Hyderabad University, Hyderabad University itself. The other one is from Hemant Reddy. He has published several papers from Texas stating that, down regulation of VDAC activates cardiolipin. And actually, the phospholipid biosynthesis takes place only when VDAC is active in the granules that are associated with these things. Unless these things are corrected, we will not be in a position to select the targets. Otherwise, it will be like what is called, as Professor Ram Reddy has stated, that we will be just searching for answers for those things. Chemically, perhaps all your questions are right as a chemical science, but from biological and therapeutic side, unless you correct your biology, we are not going to correct select proper targets.
Yeah.
Thank you for giving me this opportunity.
Yeah, thanks for your insight, sir. I think there are few questions from the participants. Once again, thanking Lakshmipad sir, and we'll go into the questions. Like,
Yeah.
I have a question here, like going to the first question. So we have been discussing about drug design. So when you talk about drug discovery or drug design, we are talking about small molecules. So one of the participants raised a question here that now you are slowly moving towards peptide design, small peptide design, and you're talking about developing antibodies. In the pandemic situation, we have seen the need of an antibiotic. So, antibody. So his question is, can we use these computational approaches to design such molecules? Like, is there any special requirement for that?
Shall I answer the question?
Yes.
Yes.
Go ahead.
Okay. So for peptide, one of the things people should consider is that often people like to build peptide which are complementary to the target protein. I think often for cancer targets, I mean, we don't know the target. I mean, how they cross the cell wall, that is a challenge, and there are many natural peptides there. So one of the challenge computationally is that how one can classify whether they are anti-cancer or for some other disease, because there are already something there. Now from the database, one can classify them. And also people can use generative AI, and the challenge over is that how to use the non-standard amino acid and certain functional group at certain position, because this functional group can actually increase the selectivity of the particular peptide that is going on.
Okay. Ram Reddy sir, you would like to add something?
Yes, the computationally, we can use pretty much the same methods. So actually, when you go for peptide, based on what is the size, how many amino acids are there. If there are only few amino acids, there's no problem. If the larger the peptide, then you are increasing the conformation flexibility and all those things. That is going to get more complicated. If it is small peptide, I don't see any problem. You can use whatever the methods we are using. That will have the pretty much same approximations.
Yeah.
So other than that, it is pretty straightforward.
Yeah. Bhartam sir.
Yes, it is very much practical, and we have done it very well in the case of kinases. In the case of kinases, the 3D structure of the substrate binding domain is actually a groove, not a pocket. Instead of being a pocket, it is a groove. To the groove, the sequence of amino acids can fit very conveniently. And then that has got a requirement also. There is a required motif. So the biology friends already suggested that a particular motif is essential for any peptide that you are going to design in the future. If that is the case, that kind of clues we take from the literature, and then we design almost thousands of peptides, and then dock all of them in the groove, not in the pocket.
Yeah.
Because the peptides are long chains. So we have to try to fit them inside the groove. And then as a result, we will possible to design, and many people have done it, for not only anti-cancer, mostly anti-cancer also on the kinases. They have worked for the peptides.
Okay. I think even the main one of the most important thing is even the parameters won't be a challenge, because the peptides already have their parameters. But when we will be designing an antibody, I think parameters are going to be one challenge, because we have glycoproteins. I don't know if there are enough parameters for glycoproteins.
Well, if you take the parameters of the amino acids, then that may fit anyway.
Yeah.
If you want to have a separate set, just do it.
Yeah. Going on to the next question, like one of the participant has posted that he's currently pursuing chemical engineering, and he's very much interested in molecular modeling as well as applications of artificial intelligence in drug discovery. And he asked for suggestions on what are the career prospects and how he can prepare himself to be industry ready.
Well,
Dr. Arjit, yes, Bhartam sir, yeah.
Yeah, I would like to tell that whenever you want to enter into a field, it appears that there are already many players in the field.
Yeah.
So it is whether to enter or not to enter, is it easy or is it difficult? It is one question always bothers anybody who is switching from one field to, if not switch, try to adopt another field. Then also these questions come into picture.
Yeah.
There is nothing wrong. But at the same time, I would like to tell the bottom of the pyramid is already occupied. So many people are working, maybe. But the moment you go a little bit the next step, the number of people who actually can contribute are relatively less. And then if you go to the next further leg,
I think there's some connectivity issues. Arjit sir, can you unmute, please?
Yeah, I completely agree with what he was saying. So that should be my answer. So my basic answer to everyone, technology is not important. Understanding the domain knowledge is the most important part for for solving any problem. Technology one can pick up in quick time, whether it's molecular modeling, AI, or something else. So that's not so difficult.
Yeah. And do we need good coding skills, sir, for AI?
Of course. For AI, you need some sort of coding skill because you basically use those type of things. So coding is of course required for these things.
Yeah.
But even for MD simulation, if you are not only using software, you need to think about some sort of coding skill. For example, during my PhD, I developed a method which is now properly known as transition path sampling. I mean, other people has done even before me. So I applied it for some specific cases. So I had to do some sort of coding over there.
Actually, if you know the coding, that would help you a long way. Because, if you don't know, you are using that package like a black box.
Yeah.
Even you get some number, you don't know what is the meaning of that number. So when you know the coding, what is inside, even something goes wrong, immediately you can figure out, okay, this is due to this one.
Yeah.
That would help you a lot. That will make you a more successful chemist or computational chemist, knowing code rather than using a black box.
Yeah. Yes, sir. And one more question, like one of the participants asked is, can we use these free energy perturbation methods as a filter? Like using docking as the initial one, and then using free energy perturbation as one filter in the virtual screening process. Yeah,
in the beginning, we cannot use free energy if we have the millions of molecules. So as I presented in my optimization slide, so we need to use crude methods in the beginning to get rid of a lot of junk. Later on, when you get to the final stage before you synthesize, so use free energy and rank order them and synthesize those molecules. Otherwise, using free energy for every molecule, that is almost impossible.
Yeah, so we can
use
it we can use it at the top level in the virtual screen.
Top level. After getting rid of other things, go step by step. First dock, then next minimization, then QSAR. The last one is FEP.
Yeah.
One good thing about all these methods is, once you understand limitations of each method, when you get some predictions, you know what is the probability of that is accurate.
Yeah.
So if you don't know the limitation, whatever the number you get, you believe that is right number.
Yeah.
So when you get some score using a doc, you should put some trust in that one. Is it 40% or 50% accurate?
Yeah.
So based on that, when you present to chemist, you should give, okay, probably it is going to be right. Because, if you start telling whatever you predict that is going to be in the right direction, so very often it goes in the wrong side. The next time they won't even believe you. So
Yeah.
that's why many computational chemists, they are losing their credibility with chemists. Once
Okay.
you lost your credibility in that company, it is very hard to bring them back.
Yeah. Yes.
So even if you use the free energy calculation, you should tell them probability is very high, it is going to be the right direction. You will never say this is going to be the right,
Yeah.
100%. There's no
Yeah.
100% guarantee for any prediction.
Yeah. So we should modestly use the word it will. Like, no.
Yeah, most likely, we never say it is 100%. The moment you say 100%, if it doesn't work, next time that guy doesn't even believe you.
Yeah.
Apply.
Yes, sir.
Yeah, to answer the question, that one of the participants raised.
Yeah.
You are the best example.
Mhm.
You started as a biologist, and then now you are a computational biologist, like any on par with anybody in the world. No, at
Yeah.
least. How was this transformation has taken place? How difficult it was? You could probably share with all the participants so that it's the path can be followed by others also.
It was tough, but I had a cooperative supervisor. So I did my PhD with Professor Reddy, and I had the opportunity to learn all the, we had Professor Rami Reddy as visiting faculty at University of Hyderabad at that time. So it was very easy. It was easy for me to approach all the experts because I was in University of Hyderabad. And I used to shuttle a lot between life sciences and chemistry. And there was a lot of, knowledge. I don't say exchange of ideas, or it was more of getting the ideas. I used to go and attend all the pre-PhD seminars of computational chemists and computational biologists. And I attended maybe around 20 conferences in my PhD. So I used to go and look at every poster and try to get an idea of, okay, how people are using, what are the approaches, and how I can fill in the gap. So it was not easy, sir, but at the same time, it was not difficult because I didn't have a flair for experimental work. Somehow my temperament couldn't, handle the experimental work, and theoretical was, it was close to my heart. And so it was a little easy for me. And the other thing, and Bhartam sir, once I was in Dharamshala, and because he's one of the pioneers in ligand-based drug design, so I used to approach Bhartam sir whenever I need help in ligand-based drug design. So it was not easy, but I met the right people at the right time, so it was easy.
I think more important.
May I also add something?
Ajit.
Yeah, if I can just add to Professor Reddy's comments. Mostly for the students on this call, the future is always going to be cross-functional, okay, interdisciplinary, interdepartmental. So please don't focus on, if you're a biologist, just don't focus on biology alone. AI is going to be, impacting everything in a big way.
Yeah.
There are lots of courses for, coding. Please look into that. Try to look into some chemistry so that, you are much more informed. Not just in your own field, but beyond that. And there is scope for it. And there's lots of courses that are available. And, there are experts here who will be able to help you. So please think multidisciplinary instead of just, being a biologist or a chemist or a computational person. Thank you.
Yes. Yeah, I was about to add, it's not interdisciplinary, it's multidisciplinary right now. And all the students right now, you have your online courses. You can enroll. What COVID has done, it has brought everything together. Before there was a jinx that can we do an online course? Can we sit through the course? But now COVID has shown us that, we can sit at home, we can work from home, we can study from home. So all the top universities, they give online courses, and you can hear all the top-notch professors sitting at home. So I think all the students can avail this and, work towards being multidisciplinary and be industry ready. And that's like what Arjit sir was saying, you must be a package of all, you must have your domain language. Then if the industry has to take you, you must know the computational chemistry things, and coding is must for even biology guys these days. Without, data analytics is the future. If you don't know data analysis, I think even you won't get an experimental postdoc. So that's going to be the future.
Sir, may I also add a few lines?
Yeah.
Yeah. So don't think the only upriser has that luck to work under Reddy sir. even you all can work under Reddy sir. So Reddy sir has built Faba Academy. So with the help of Ajit and others. So Faba Academy is all about helping people, whatever doubts you have, especially in the life sciences. So it's not only Aparai sir, you also have the opportunity to work with Reddy sir. That's it. Yeah.
And before we close, maybe I had one question, like which I usually many students ask me, and then I thought the experts can give an answer for them. Like we know that these docking programs, we have been discussing that when we use these rapid-paced virtual screening methods, and one of the most widely used rapid-paced virtual screening method is docking. It's quite close to all the internship candidates, because the best internship they want to do is they want to come to our lab, they just want to use docking, they'll get instant results and they'll go. So on a serious note, when we know that these docking programs are not so accurate, but we know that it's a fast screening process and it helps in screening of large chemical databases. So my question is, how should we see this? Should we see this like a glass half empty, or should we see this as a glass half full? How we should see this scenario?
Because if anybody believes that the doc program can predict something, they can say it's a glass half full. Anybody doesn't believe that it's glass half empty. So, but I do believe that there is some value, particularly to get rid of a lot of junk out of the millions of small molecules.
Yeah.
So once you go to the final predictions, so it has its own limitations, and we should not put a lot of weightage on those predictions.
Yeah. Bhartam sir.
Yeah, I would like to say that this question can be asked everywhere. Maybe very, very colorful sciences, very, very attractive sciences also, we can ask the same question. So many of the people working in our field, drug discovery, they trust the organic chemistry reactions are much more trustworthy, and computational methods are relatively on the weaker platform. It is not correct. It is true. Every science has got its own ups and downs. There are, especially if I want to refer to organic chemistry, I think there are about more than 1 lakh organic chemistry people. Not all of them are great organic chemists. We cannot, we know Woodward was great organic chemist, but does not mean that every organic chemistry is great. So likewise, in our field also, everybody has to struggle. Everybody has to bring out the value to the point. And everything has got an art. There is an art and science. If we can mix this art of doing science, then we will succeed. If we do science as only as a science, we will fail. So there is, just like a musician becomes a great by practicing. There is a Telugu song, there is a Telugu poem says that that's what we have to do, whether it is organic chemistry or whether it is computer modeling, whether it is physics or rocket science. Everywhere there is art as well as science. If we mix them, result will be guaranteed. We have to be very ready to receive the right answers in the right time.
Ajit sir.
I think it's time to improve the docking method, and I think I have seen one work which has significantly tried to improve the docking program, which is known as GNINA.
Yeah,
I think many of you see. So basically, what it used, it trained the model. It's also a AI-based method. It trained the model based on the existing conformation, and then for a molecule, so you have number of model docking scored method. Say let's say top 10. Now, instead of ranking them based on those docked scoring method, it tried to use a different scoring function, which they call as a binding energy. Now, these binding energy is more accurate than the docking score. So I think within few years down the lane, I think there will be more such work that will try and come, which can actually give you, because instead of your best docking score, maybe your fifth one has a better binding energy. So those type of things will come as we move forward.
Yeah, I think I'd like to add 1-2 sentences from my side. Like most of these docking programs, because the scoring functions that are built are trained. These are trained models, which are you take numerous drug targets and we train and we get the scoring function. So basically, the user also must have thorough information of what kind of interaction plays very crucial role in that particular drug target. And they must know how to play around with these scoring functions, how to use soft potentials, how to use the partial charges in the protein. So that when you can modify these and you can fine tune the docking program. It's not that docking program will become as accurate as your molecular dynamics based methods, but at least you must learn how to fine tune them. You must have thorough knowledge on your drug target, and you must learn how to fine tune them. That's one of the most important. You must try to make the best of what you have. How to modify it and how to give value to it. That's what you must learn.
I think one more point is once you understand the limitations of the docking, I mean, what are the in theory what is missing in the equation?
Mm-hmm.
So if you start incorporating those whatever the missing parts, eventually down the road you may get better agreement.
Yeah.
Because where they started in 1980s and now there's a big improvement.
Mm-hmm.
So if you go to for the next 10 to 20 years, then the docking may play a very important role.
Yeah.
The thing is, we need to get rid of those approximations one at a time. It takes a long time. It's not going to happen overnight.
Yeah.
But at the end of the day, you need to do some sort of free energy calculation.
Yeah.
Understand what is better.
Mm.
Yeah, but it is very hard to do for the whole databases.
Right.
But
No, only for the top 10, top 100, 1,000.
Yeah.
Yeah.
But still the docking plays very important role in the early stages of screening. So we should not say that it's useless. Yeah.
True.
Yeah. Any more questions from the participants? Let me just check if I'm missing anything. So no more questions.
Select the targets properly. That is the only thing. And that selection of properly, put your metabolic pathways properly. Don't go blindly by Warburg effect or TCA cycle. These two models have destroyed 100 years of science. Without hesitation, I tell this.
Yeah.
I completely agree with you.
If you take pyruvate metabolism itself, there are 5 enzymes which are actually modulated. Pyruvate kinase, pyruvate dehydrogenase, blah, 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 like that. Each one has different roles. And the same molecule, same output. Phosphoenol pyruvate, once it was considered to be the product of ATP, now becomes what is called the product of actually the phosphorylation techniques. So unless these things are corrected, we will not be in a position to put this thing. Proof of pudding is in eating it. We have millions of molecules identified, and then they end up in clinical trials. Thank you very much. Thanks for giving me the opportunity, Dr. Redana, and also Dr. Aparoy.
Yeah. Sir. Thank you, sir. Thanks for your insights.
Thank you, everyone. Especially Rami Reddy Mutyala garu, it's 3 a.m. for you in the morning.
Yeah.
So I think that the commitment to teach, commitment to contribute to others, it's commendable. Thank you so much for joining. And also Lakshmipati garu, Professor Lakshmipati garu, thanks for joining. It's like you are challenging everybody not to just follow textbooks, but also think outside the box. So that's commendable. So I would like to thank everybody. Yes, sir.
What has happened is 100 years of this Warburg effect has challenged 500 years of earlier science. That is the major problem.
Yeah.
I mean, the ability to challenge that the way to think is something that people have to learn from you, sir. Otherwise, we just take things for granted and we follow it.
Yeah, he has a lot of ideas. I think the only thing is we have to spend and understand those things. Otherwise, you ask him, he's going to contribute a lot for everything.
Yes, Dr. Reddy, the slide which I have shown, glycerol D3-phosphate dehydrogenase, which is considered to be the Warburg enzyme, is critical in partitioning actually the ATP production, nucleic acid production, and also gluconeogenesis program and then, modulating carboxylations where vitamin B component roles become important in this thing. If you examine that thing, I am prepared. If you are in Hyderabad, I am prepared to discuss that in detail.
No, you always throw a lot of ideas. So you are very good. So thank you for
So-
those ideas. Yeah.
So Professor Lakshmipati is the one who learned all these after retirement.
Yeah.
Oh,
Still he is very active.
After
Now, these
retirement,
questions were there in my mind, but
Yeah.
internet provided
So-
me an opportunity to look into the 100 years
So,
of literature and
after
beyond.
joining Cellworks in Bangalore, he mastered the entire signaling pathways. And then today, he is the expert in all the signaling pathways, specifically metabolic pathways, and challenged many of the age-old concepts, including the Warburg effect, everything. And in fact, I strongly suggest some of the students to go through his articles, which were published in all the leading journals. And I must thank Rami Reddy again for sparing his time. Whenever he is in Hyderabad, he always interacts with the students, and that's how he trained. Many of the students from my lab, specifically on computational skills, because I didn't have any knowledge on that. It is only Rami Reddy who trained many of our students. And Professor Bharatam, of course, we know him since a long time, and Rami Reddy, along with Professor Bharatam, we conducted several workshops on molecular modeling and drug design, and he was always ready to participate whenever Dr. Rami Reddy is there. And Dr. Roy also we interacted earlier, and I think we need to interact much more now, having known you, being our neighbor. I think we will definitely invite you to the campus and then interact with you. So
Yeah, so I often visit campus. I have two collaborators.
please come to Life Sciences.
Yeah, so
Life Sciences,
Professor Minal and Shubhra, so I worked with several papers, 3 papers with them. We did the simulations.
Jagadish, you can conclude now.
Basically, I would like to conclude here. So thanks everybody, especially the participants whoever came here, and of course the speakers. Rami Reddy garu, I think you can sleep again now.
Okay.
You go now.
So once again, I thank you organizers for giving this opportunity. Yeah.
Sure, sir. Yeah.
Okay.
Thank you.
Thank you. Thank you.
Thank you.
Okay, sir.
Thank you, everyone.
Thank you all.
Bye.
Thank you all.
Thanks.